We are here with a quick, rapid revision of science and technology concepts through the PYQs, getting into the topics one by one. So first we are going to see the biotechnology based questions that were asked in science and technology. Any suggestions, any doubts or clarifications, or if you want me to take any particular topic of science and tech to make things better or easier to understand, please do let me know in the comment section. Hello everyone, welcome to PWL IAS. We are here with a quick, rapid revision of science and technology concepts through the PYQs. Without any further delay, taking you right into it. But before that, this is my official Telegram channel. Any doubts or queries that you have, you may ask it over here. You may also send in your PYQs from 2013 onwards. That is the mains PYQs from 2013 onwards in the PDF format of the optional public administration, all the 4 GS, and essay. Clear, people? So let us start getting into the topics one by one. So first we are going to see the biotechnology based questions that were asked in science and technology. Consider the following statements. Biofilms can form on medical implants within human tissues. Biofilms can form on food and food processing surfaces. Biofilms can exhibit antibiotic resistance. What's a biofilm? Generally, these are made of microorganisms. It can be bacteria, it can be a virus, anything it can be. Number one. Number two, it's like a layer, thin layer of surface coating it will be. And yes, they are resistant to antibiotics. Alright. They can form on food as well as food processing surfaces. They can also form on medical implants. They can form on any of these three. So basically, wherever you will have these polysaccharides means these are made of carbohydrates. They are like a glue-like structure there. Glue-like structure means it sticks on. So it provides a shape or structure. So you will find it in animal tissue, you will find it in plant tissue, you will also find it in biofilm. Clear? So it can form both on, biofilms can form both on living tissues as well as non-living tissues. It may also contain impurities, some minerals, some corrosive particles. All this you can find it on a biofilm. Alright. What is the answer then? D is the answer. Let us look into the explanation here. Statement 1 and 2 are correct. Biofilms may form on a wide variety of surfaces, including living tissues, food and food processing surfaces, indwelling medical devices like catheters, heart valves, etc., human and animal tissue, industrial or potable water system, piping or natural aquatic system. As biofilms attach to each other and to the surfaces, they are capable of acting as barriers to antibiotics. Clear, people? A biofilm is a collection of microbial cells that are encased in a polysaccharide-based matrix and are permanently attached to a surface. That is, it cannot be removed by gentle rinsing. Polysaccharide matrix, like I told you, it's like a glue-like structure. It provides rigidity, strength it provides. Moving on. Van Leeuwenhoek, using his simple microscopes, first observed microorganisms on two surfaces and can be credited with the discovery of biofilms. Microorganisms that form biofilms include bacteria, fungi, and protists. Non-cellular materials such as mineral crystals, corrosion particles, clay or silt particles, or blood components, depending on the environment in which the biofilm has developed, may also be found in the biofilm matrix. That is, obviously, piping system means you are going to find some corrosive materials in it or some mineral materials in it. If you're talking about a medical device, you're going to find blood in it. Correct? Depends upon which environment it is in. Alright. Moving on. In the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following statements. The Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform. Sputnik V vaccine is manufactured using vector-based platform. Covaxin is an inactivated pathogen-based vaccine. The first part of the statement is correct. The second part is wrong. What is mRNA platform? See, messenger RNA, that is what mRNA means. So, in my body, any protein has to be manufactured. How it is done? So, whatever information is there, the genetic information, whatever I have, that is present where in a DNA. Whichever protein that needs to be made for the body, that is present in the DNA. From the DNA, that whatever is the coding for manufacturing the protein, that has to be copied. That copying work is done by whom? Your mRNA. And that is then transferred to where? Ribosomes. Getting the point here? Any protein that has to be manufactured in my body, the coding of it is present where? Inside DNA. That is then copied. By whom? Messenger RNA. Then from messenger RNA, the information goes to the ribosomes, where the protein is finally synthesized. Clear? Statement 1 is incorrect. The Serum Institute of India produced the COVID-19 vaccine Covishield, but not using the mRNA platform. Instead, it was developed using the viral vector platform. What do you mean by a viral vector? Vector means carrier, viral means virus. So, I need to put the COVID-19 virus, not in its full form. Maybe a part of that virus I will take. Then I will put it in some other virus, which is not harmful for the human body. Then I inject it into the human body. That is vaccination. So what is the viral vector platform here? What vector have we used here? The vaccine uses a chimpanzee adenovirus, ChAdOx1, which has been modified to carry the COVID-19 spike protein into human cells. Spike protein simply means this is how it will look. Like a spike. That part I will take, I'll put it in this chimpanzee's virus and then inject into the body. So it will not cause much damage here. And best part is, it will help in developing immunity against COVID also. This cold virus is incapable of infecting the recipient, but it effectively trains the immune system to develop a defense mechanism against similar viruses. That is the best part here. You need not actually inject the full virus into the human body. Only a part of it is enough. And the body will develop antibodies for it and develop the immunity for it. So the next time when the actual virus comes also, the body is ready. The same technology has been used to develop vaccines for viruses like Ebola. Sputnik V, the world's first registered COVID-19 vaccine, is based on human adenoviral vector-based platform. This technology, the gene from the adenovirus that causes infection is removed, and a gene encoding a protein from another virus spike is inserted. Human adenoviral vector-based platform. What does it mean? Adenovirus. Common cold virus. Common cold, sneezing, coughing, etc. We will get it right. That is taken. That virus is taken, which is not harmful for us. And then, from that, the gene from the adenovirus that causes infection is removed. So if I inject again common cold virus directly into your body, what will you get? You will start sneezing. You will get cold again for a week. I don't want that. So whatever will cause common cold infection, that part I remove. Then into this, what I am adding? A gene encoding a protein from another virus spike. Means I am putting that same protein spike from corona. Getting the point here. Earlier what it was? Chimpanzee. Chimpanzee's virus into that protein spike is injected of corona, then that is vaccinated, correct? That is viral vector platform. Here, human adenoviral, this is also vector based. Here also we are having a carrier, but this is not from any chimpanzee or any animal. This is our normal common cold virus. But from that, whatever will cause infection, that part I remove. What other part I will put in there? Again the protein. Clear? Statement 3 is correct. Covaxin is an inactivated viral vaccine. It is developed using the whole virion inactivated virocell-derived technology. This means the vaccine contains inactivated viruses that cannot infect a person but still train the immune system to build a defense mechanism against the active virus. Means what? Either using radiation or high temperature or maybe chemicals also. Sometimes the whole virus is made inactive. That means if I inject the virus into your body also, it cannot multiply or reproduce. So here when I inject it into your body, what happens now? Again, as far as the body's immunity is concerned, it is a foreign object. It will develop antibodies and it will remember this. And based on that, when the next time the real active virus comes again, the body is ready to fight it out. Clear, people? Hope this was understandable. Aerial metagenomics best refers to which one of the following situations? Collecting DNA samples from air in a habitat at one go, understanding the genetic makeup of avian species of a habitat, using airborne devices collect blood samples from moving animals, sending drones to inaccessible areas to collect plant and animal samples from land surfaces and water bodies. Correct answer is option A. That is, metagenomics delves into the study of microbes within their native habitats. Oh, you use aerial technique to collect the entire virus sample from a given area. Aerial metagenomics entails collecting air samples from a specific setting to capture the genetic material of various microorganisms like bacteria, viruses, fungi, and any other microscopic life forms. Clear, people? Moving on. Microsatellite DNA is used in the case of which one of the following? Studying the evolutionary relationships among various species of fauna, stimulating stem cells to transform into diverse functional tissues, promoting clonal propagation of horticultural plants, assessing the efficacy of drugs by conducting series of drug trials in a population. Microsatellite DNA. So we know in DNA what do you have? AT, GC, pairing. Correct. Sometimes certain sequences will be there. Microsatellite DNA, when we talk about, it talks about those sequences in the DNA that repeats. So how many times that sequences repeat? Say for example, TAC, TAC. Alright. How many times this repeats? Depends highly on what? Depends highly from individual to individual. Depends upon the genetic diversity. In other words, your genetic diversity or your uniqueness, etc., can be determined using what? Microsatellite DNA. How unique that coding is. So this can be utilized even in paternity tests to find out about certain genetic diseases or certain unique traits, etc. To study about it, this field is very, very good. Let us look into it in detail. Microsatellite DNA, alternatively known as short tandem repeats, represents a genetic marker characterized by repetitive sequences of DNA. That is the main highlight here. How many times it repeats? Highly polymorphic. Highly polymorphic means what? It varies from individual to individual. So sometimes certain uniqueness also within the species, outside the species, you will be able to detect because of this. Case in point being, now many years ago, a study was conducted by some university, and they published an article telling that there are very high chances that Aishwarya Rai, who is a Bollywood heroine, and Angelina Jolie, who is a Hollywood heroine, both of them probably could be from the same ancestor. Why? The unique eye color what both of them share. First of all, it's a very rare color, and both of them have very similar colored eyes, if not the same. And that is only possible if their ancestor is the same. Getting the point here. This is what you call polymorphism, the uniqueness of a DNA. Again, within the species also, there will be uniqueness. Between species also, when you cross compare, there will be some changes. Clear? Microsatellites exhibit varying lengths attributed to discrepancies in the number of repeat units. By scrutinizing the discrepancies in microsatellite DNA sequences within individuals or population, scientists can evaluate genetic diversity, population structure, and evolutionary links both within the species and across different species. By looking into the number of repeats, why some are having 3, why some are having 5, some are having 6, some are having 4, etc. Looking at the number of times this is getting repeated, scientists can come to a conclusion. All right, people. With reference to recent developments regarding recombinant vector vaccine, consider the following statements. Genetic engineering is applied in the development of these vaccines. Bacteria and viruses are used as vectors. Which of the statements given above is or are correct? 1 only, 2 only, both 1 and 2, neither 1 nor 2. Again, recombinant vector vaccine. Recombinant. That means, imagine it like this. I need a harmless carrier to carry a part of the disease causing virus, only a part. Getting the point here? So find a harmless carrier, put the part of the virus into it, and then I inject it into the body. That is your recombinant DNA technology in simple words. As we can clearly see from the statement itself, both the statements are correct. Let us look into the explanation here. Are live replicating viruses that have been modified to carry additional genes taken from a pathogen. These additional genes produce the proteins we want to elicit immunity against. Additional genes from a pathogen. And they are actually nothing but what? Proteins. So same thing again. That spike protein carried in a common cold virus. Same thing. Statement 1 is correct. The techniques of genetic engineering, including creation of recombinant DNA, use of gene cloning and gene transfer. Recombinant vector vaccines expose the body to proteins made by a virus or bacteria and are often made by using weakened or inactive versions of that virus or bacteria. That is how it is. Again, what you are taking is an inactive version or a dead version also it can be. You insert it into another virus which is not harmful. Any virus it can be. Chimpanzee's virus it can be. Common cold virus it can be. Getting the point here. And then you inject into the body to elicit the same level of response when actually exposed to the real virus. Alright. This is just a diagrammatic representation of it, explaining the whole thing. You may just refer it. Consider the following statements. 2020. Genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. Person's genome can be edited before birth at the early embryonic stage. Human-induced pluripotent stem cells can be injected into the embryo of a pig. All the three statements are correct. Genetic changes can be introduced in the cells that produce eggs or sperms means that is basically nothing but germline therapy. Germline means germ. That is very small, tiny. Even you can make changes to the ovary or even to the sperm. The next stage is what? As an embryo can you make change? This was done in China. Where a mother was pregnant with twins. And there was a chance that or a probability that the twins may have inherited a certain genetic rare disease from their parents. So the doctors, what they did, they altered the DNA. They did genetic engineering. They did gene editing to both the babies while the babies were still inside the mother's womb. So this has already happened. This is actually very old news. Very, very old news it is. I mean, I know it is 2020, but the news article, what I am referring to, that had appeared at least somewhere in 17 or 16. Can be edited before birth at the early embryonic stage. Human-induced pluripotent stem cells can be injected into the embryo of a pig. With regard to pig, again, let me tell you another thing. Pluripotent stem cells can be put inside a pig's womb to grow different organs or parts. Number 1. Similarly, it has also been found that pig's skin and liver, etc., other organs also can be used for organ transplantation with a human being. So pig skin, etc., again it is cut out and it is used for skin grafting, especially for burn injuries, acid injuries, etc. Pig skin can be used. Now, normal way of doing it is what, on the face or somewhere if any injury is there, they generally tend to take the skin from the thigh. What if the thigh itself is damaged? Getting the point here. So you look for alternatives. So such methods are being employed very widely these days. So the answer to this is also yes. All the three are correct. In germline gene therapy, DNA is inserted into the body's cells that make eggs or sperm. The reproductive cells. This kind of treatment enables the repair of disease-causing gene mutations that are unavoidably inherited from parents to children. That is, either it can insert it into whom? The mother or to the father. Statement 2 is also correct, although germline gene therapy is prohibited. It is currently prohibited because obviously why it is prohibited? can actually lead to creation of superhuman soldiers. That is the main problem there. Number one. Number two, it can lead to this whole concept of designer babies. I want my babies to have this particular style of hair, this particular style of skin color, etc. Such things can happen because whoever has the money, they will try to do all sorts of nonsense, right? That's the reason why germline gene therapy is prohibited. It is conceivable to alter the genome of an embryo to prevent the vertical transfer of specific diseases and medical disorders. The example that I told you about China. Statement 3 is correct. To conduct an experiment and determine which type of human stem cell would survive the best when injected into pig embryos. Intermediate human pluripotent stem cells were the ones that endured the longest and shown the most potential for further development. What do you mean by pluripotent stem cell? What is stem cell first of all? Stem cell, imagine it like clay. With clay, you can make any structure. You can make a ball, you can make a statue, you can make anything that you want. Just like that, using a stem cell, I can develop brain tissues, nervous tissues, bone, cardiac muscles, liver, kidney, skin, anything I can develop. What do you mean by pluripotent stem cells? Pluripotent, when we say a stem cell can be developed into any type. Any type, any cell you can develop from it. What do you mean by intermediate human pluripotent cell? There are 3 stages basically. One is naive. The other is primed. Naive is like it's the most basic form of a stem cell from where you can develop it into any form you want. Primed means these are little more matured. That is, they can only be converted into a specific form. That is, maybe if they are only for, they can be only for cardiac, means only cardiac. From those stem cells, you can only develop cardiac, means you can only develop cardiac muscle and tissues. They can only be developed for nervous tissue, means they can only be used for that. You cannot develop anything else out of it. Excuse me. Your intermediate actually comes in between. They are neither too naive nor primed. You can use it for whatever purpose you want. Clear? In the context of recent advances in human reproductive technology, pro-nuclear transfer is used for fertilization of egg in vitro by the donor sperm, genetic modification of sperm-producing cells, development of stem cells into functional embryos, prevention of mitochondrial diseases in offspring. That is the right answer. Pro-nuclear transfer or preventing mitochondrial disease in offspring. So father will give the sperm, mother will give the ovary. Correct? From that, what you get is a fertilized egg. From this, what I remove is the nuclear DNA, or from the mitochondria. That portion I remove from this. Alright? Because why am I removing this? Because this contains all the genetic material. Your mitochondrial DNA contains all the genetic material inherited from your parents. But one of them, let us say, is carrying a rare genetic disorder. I don't want it to transfer to the baby. Now what do? I take another egg of a donor, which also will have what? Their own nucleus. Okay, I remove this. And then what do I insert? I insert this nucleus here. The baby will largely have whose DNAs? Both the parents' DNA. Largely. But will not inherit any of the diseases. Getting the point here. This is father and mother's. After fusion together, egg is formed. From this, I remove this nucleus. And into the donor egg, I insert it. Let us see it now. Mitochondrial transfer methods employ proton transfer. In pronuclear transfer, a zygote is first created. Zygote is nothing but an egg. Zygote is created by first fertilizing the mother's egg with the father's sperm. So you create a fertilized egg first. Sperm and ovary put together. The pronuclei of the egg. What is that? Pronucleus is the nucleus of the egg or sperm at the stage of fertilization prior to the nuclear fusion. The nucleus and sperm are then removed from the zygote and inserted into a donor egg, which has been fertilized and has its own nucleus removed. I know they have wrote the whole explanation in a little confusing manner. That means in the donor egg there is going to be no nucleus. All right. But so where will the nucleus come from? It will come from the parents' fertilized egg. From there I will take it and put it. That's it. Donor egg which does not have any genetic disease, I will remove its nucleus. Nucleus I am taking it from the parents. That's all. The zygote derived from the donor egg is then inserted into the mother's uterus. So it goes again back. Now, after putting it, what happens? You take the whole thing back to mother's uterus again, not to the donors. The pronuclear zygotes produced from donated oocytes are extracted from a karyoplast using micro-manipulation equipment and thrown away. Zygotes, when we say, these are again egg only. Pronuclear zygotes produced from the donated oocytes. Donated oocytes means the unfertilized eggs are called, or immature eggs are called oocytes. So, produced from donated oocytes are extracted from a karyoplast. Karyoplast is again nothing but the nucleus. You extract it out and then it is removed and thrown away. Because you don't require the donor's nucleus there, right? You are just extracting it out and you're throwing it and you're replacing it with what? The father's and the mother's nucleus. Two pronuclei from the afflicted zygote, also in the form of a karyoplast, are transferred into the enucleated healthy zygotes during a therapeutic pronuclear transfer. The resultant zygotes have mtDNA from a donor and nuclear DNA from each of the intended parents. That means the nucleus present inside the nucleus, the mitochondrial DNA will be of what? Largely the parents only. But mtDNA will be there from a donor. Clear? Two pronuclei from the afflicted zygotes are transferred into the enucleated healthy zygotes during a therapeutic pronuclear transfer. The resultant zygotes have mitochondrial DNA from a donor, obviously, because that is coming from the donor only. And nuclear DNA from each parents, because nuclear is from the parents. That is, nucleus is what we are replacing. Clear? What is Cas9 protein that is often mentioned in the news? Very direct question. Molecular scissors used in targeted gene editing. The Cas9 enzyme functions as a molecular scissor that can cut DNA, and the single guide RNA contains a sequence that can bind to the DNA. Together, these two components make up the genetic tool. That is very simple. Let us say you have a DNA structure here. Okay. And in this one particular region, there is a faulty DNA. So what I do is this RNA, guide RNA will take me to this faulty area. Alright, once it reaches there, what happens next is the Cas9 enzyme will cut it here. And after it undergoes cutting, then what happens? It starts healing. It slowly starts regrowing back. But once it starts regrowing back, it will not have its earlier genetic defect. Finally, that defect won't be there. So first, your RNA will tell you where to go and cut it. You cut it there. And after that, what happens? It slowly starts regrowing back. Once it regrows back completely, the genetic defect gets taken care of. Clear, people? About CRISPR-Cas9, clustered regularly interspaced short palindromic repeats, or CRISPR-Cas9 is the abbreviation. With the use of CRISPR genome editing technology, researchers may quickly and precisely alter any genome's DNA. As a sort of adaptive immunity, bacteria use the CRISPR-Cas9 system to identify and eliminate viral DNA. Researchers may add, delete, or modify specific DNA sequences from the genomes of higher species using the CRISPR system's components. The Cas9 enzyme functions as a molecular scissor that can cut DNA, and the single guide RNA contains a sequence that can bind to DNA. Together, these two components make up the genetic tool. So, you must understand only one thing here. Addition you can do, deletion you can do, or maybe simple alteration. Clear? Biorock technology is talked about in which one of the following situations? Restoration of damaged coral reef, development of building materials using plant residues, identification of areas for exploration or extraction of shale gas, providing salt licks for wild animals in forests or protected areas. Answer is option A, coral reef restoration. The Zoological Survey of India, with the help from Gujarat Forest Department, is attempting for the first time a process to restore coral reefs using biorock or mineral accretion technology in the Gulf of Kutch. Biorock is the term used to describe the material created by the electron accumulation of minerals dissolved in salt water or steel structures that are lowered onto the sea floor and connected to an energy source, in this case solar panels, that float on the surface. That is basically you are ensuring a steel structure you create and you place it inside the sea. Why? So that coral reef can be regrown. That is the idea behind it. Biorock technology it is called. Consider the following statements. DNA barcoding can be a tool to assess the age of a plant or animal, distinguish among species that look alike, identify undesirable animal or plant materials in processed foods. Which of the above statements is correct? 1 only, 1 and 2, 3 only, 2 and 3. Statement 1 is incorrect. Scientists use a radioactive carbon isotope called carbon-14 as their clock to determine the age of plant and animal remains from the more recent past. DNA barcoding is a taxonomic technique that identifies an organism as belonging to a specific species by using a short genetic marker in its DNA. As you can clearly see, this is almost similar to our microsatellite DNA. DNA barcoding simply means what? Getting the code over there. The DNA coding there, and based on that coding you get to know about the genetic variation or genetic variety of species within or outside the species. An individual DNA sequence is compared to a reference library of similar DNA sections to accomplish this, called sequences. So if I have the DNA sample, I can cross compare and check and then I can verify it. Again, a methodology that is used even in forensics to find the convicts, to find the suspects as well as to prove paternity suit, etc. You can use this. Statement 2 is correct. DNA barcoding helps in the identification of species even from minute amounts of tissue, which can help tackle problems like illegal trade of endangered species. DNA barcodes can effectively control pest attack, identifying disease vectors properly. That is, you find out what is the reason which type of pest is attacking the crop. And what do? You insert the counter DNA into that crop so that the crop becomes pest resistant. Using DNA barcoding, which is widely used in fish species, is a viable method for authenticating the processed food. Using DNA barcoding, natural resources managers can monitor illegal trade of products made of natural resources like hardwood trees. DNA barcoding can be used to identify important medicinal plant. That is, you can check the authenticity. Getting the point here. When somebody says, no, this is not wood, you will get to know whether it is wood or not. Similarly, when somebody tries to say, no, this is not an endangered species, you can actually find out whether it is endangered species or not using DNA barcoding. Bollgard 1 and Bollgard 2 technologies are mentioned in the context of clonal propagation of croplands, developing genetically modified croplands, production of plant growth substances, production of biofertilizers. Both again keeps popping up in the newspaper. Correct answer is option B. Bollgard 1, single gene technology, is India's first biotech crop technology. B means developing genetically modified crop plants. Approved for commercialization in India in 2002, followed by Bollgard 2, double gene technology in mid-2006. Bollgard cotton has an insecticidal protein from a naturally occurring soil microorganism called Bacillus thuringiensis and offers built-in protection for cotton against destructive American bollworm Heliothis armigera infestations. The enhanced double gene Cry1AC, Cry2AB technology found in Bollgard 2 technology protects against bollworms and Spodoptera caterpillars, improving ball retention, maximizing yield using fewer pesticides and preventing insect resistance. With reference to the recent developments in science, which one of the following statements is not correct? Functional chromosomes can be created by joining segments of DNA taken from the cells of different species. Pieces of artificial functional DNA can be created in laboratories. A piece of DNA taken out from an animal cell can be made to replicate outside a living cell in a laboratory. Cells taken out from plants and animals can be made to undergo cell division in laboratory Petri dishes. A is the answer. With reference to the recent developments in science, which one of the following statements is not correct? Functional chromosomes can be created by joining segments of DNA taken from cells of different species. Statement 1, Statement 2, Statement 3, Statement 4. Pieces of artificial functional DNA can be created in laboratories. A piece of DNA taken out from an animal cell can be made to replicate outside a living cell in a laboratory. Cells taken out from plants and animals can be made to undergo cell division in laboratory Petri dishes. So let us look at the statements one by one. First statement is what? Functional chromosome can be created by joining segments of DNA taken from cells of different species. Can I take a plant gene, animal gene, etc. and make it completely functional chromosome? Can I do it? No. Anybody I can take like that? Absolutely not. That first statement itself is wrong. Combining of DNA fragments acquired from cells of different species cannot result in the creation of functional chromosomes. Statement 2 is correct. And what is that statement 2? Pieces of artificial functional DNA can be created in laboratories. Absolutely. Pieces it is, not complete DNA. Pieces I can replicate, no doubt about it. In a lab, artificial functioning DNA fragments can be created. Japanese researchers announced the creation of the first DNA molecule in the world that is almost entirely formed of synthetic components in 2007. Statement 3 is correct, which is a piece of DNA taken out from an animal cell can be made to replicate outside a living cell in a laboratory. Absolutely true. Cloning, Dolly the sheep, or even in a lab in USA many years ago, they were actually able to create a burger patty made out of cow's DNA. They didn't kill the cow for it. They extracted the stem cells from it and they developed the burger, the meat inside the laboratory. So you didn't kill anybody. Getting the point here. In a lab, it is possible to get a portion of animal DNA to reproduce on its own outside of a living cell. It is done through the polymerase chain reaction technology. Statement 4. Cells taken out from plants and animals can be made to undergo cell division in laboratory Petri dishes. Absolutely true. In a lab setting, Petri dishes can be used to induce cell division in isolated plant and animal cells. Plant cells are totipotent and a single cell has the capacity to divide through the process of mitosis in sterile conditions in a culture Petri dish to develop into a matured plant. Petri dish means these are like dishes where, bacteria, certain bacteria, etc. are kept so that cells can multiply easily. RNA interference technology has gained popularity in the last few years. Why? It is used in developing gene silencing therapies. It can be used in developing therapies for the treatment of cancer. It can be used to develop hormone replacement therapies. It can be used to produce crop plants that are resistant to viral pathogens. Select the correct answer using the code given below. 1, 2 and 4. B 2 and 3, C 1 and 3, D 1 and 4 only. RNA interference technology, otherwise it is sometimes also called as a mute technology. So earlier I told you, from a DNA, the coding for protein is copied by the messenger RNA, and from there it goes to the ribosome and then the protein is finally made. But what if I don't want the mRNA to copy everything? I don't want it to copy the genetic disorders, the rare diseases. Those certain parts of the RNA, I want it to be silenced. That is where this technology comes. Let us see. Statement 1 is correct. Its natural mechanism for sequence-specific gene silencing promises to revolutionize experimental biology and may have important practical applications in functional genomics, therapeutic intervention, agriculture and other areas. Everywhere you can use it. Statement 2 is also correct. Numerous studies have demonstrated that RNAi can provide a more specific approach to inhibit tumor growth by targeting cancer-related genes. Say for example the oncogene. These two, it is possible. What about hormone replacement? Hormone replacement means in my body, can I replace testosterone with progesterone and estrogen, a female sex hormone? Or can in a female using this technique, can I change the hormone from estrogen and progesterone to testosterone only? Can I do that? No. As of now, is it possible? Medically, is it possible to change the hormones like this? It is possible. There are some things called as hormone blocker tablets are there. That is, if I want to become a female, what do? I take hormone blocking tablets. That is, block the testosterone production in my body and start consuming tablets of estrogen and progesterone. Same way for a female also. But that you cannot do using this technique. This technique is only for what? Muting certain parts of the RNA, messenger RNA. So statement 3 is incorrect. Statement 4 is definitely correct because reverse experiment, virus-induced gene silencing, in which short sequences of plant genes were introduced into viruses, showed that targeted gene was suppressed in an infected plant. It will produce crop plants that are resistant to viral pathogens. What is the application of somatic cell nuclear transfer technology? Production of biolarvicides, manufacture of biodegradable plastics, reproductive cloning of animals, production of organisms free of diseases. What is the application of somatic cell? So what is somatic cell first of all? Somatic cell simply means any cell which is not a germline cell. That is, it is not the sperm, it is not the ovary or ovum. Any other cell other than that, any other cell other than the sperm cell or the ovary cells. All right, that is the cell. From that somatic cell, this is a somatic cell. What do I remove from it? I remove the nucleus. And what do? I already have a egg here whose nucleus has been already been removed. And this I will put it there. So what it will be? Take a guess. Option C. The most practical application of somatic cell transfer is in the reproductive cloning of farm animals that have exceptional qualities such as the ability to produce large quantities of milk. A somatic cell's nucleus is transferred to the cytoplasm of an enucleated egg using the somatic cell nuclear transfer procedure. SCNT-derived blastocyst used in reproductive cloning is implanted into the uterus of a surrogate mother, where it develops into a fetus and is carried to term. That is the point here. You take it and put it into the egg, and then it fertilizes and it comes out as a baby. Clear? The other one, so what about the other one, sir? There you didn't talk about cloning. That is not cloning simply because their mitochondrial DNA is involved. And there are 3 people involved there. There is a father, there is a mother, and there is a donor. The idea behind that is to ensure that the genetic disorders are not passed on to the baby. But somatic cell nuclear transfer, it's largely about cloning. With certain traits, I need a certain baby, a certain farm animal. To develop that, we use somatic cell transfer technology. The first mammal to be cloned by SCT was a sheep named Dolly, who was born in 96. The method might potentially be used to bring back extinct animals. For instance, frozen woolly mammoth cells could be employed as nuclear donors for enucleated elephant eggs. With reference to agriculture in India, how can the technique of genome sequencing, often seen in the news, be used in immediate future? Genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance in various crop plants. This technique helps in reducing the time required to develop new varieties of crop plants. It can be used to decipher the host pathogen relationship in crops. 1 only, 2 and 3 only, 1 and 3 only, 1, 2 and 3. Genome sequencing. Genome sequencing means, see, for each one of us there is an ATGC pairing. Okay, A, T, G, C, C, G, T, A, T, C, G, G, C, like that. This sequence varies from individual to individual, organism to organism. So once I get to know what is the correct sequence that is there, most of the genetic disorders, whatever Indians are suffering from, or most of the genetically related issues that plants are also suffering from, I will be able to find a rectification for it. Once I get to know what is the sequence, where there is a problem with the sequence. Getting the point here, people? So let us see this now. Genome sequencing is figuring the sequence of DNA bases such as A, C's, G's, and T's that make up an organism's DNA. These genetic letters total more than 3 billion and make up the human genome. So it can identify genetic markers for disease resistance and drought tolerance in various crop plants? Absolutely. I can exactly find out where it is. This technique helps in reducing the time required to develop new varieties of crop plants. Absolutely true. Because older variety is what? Maybe again, take Bacillus thuringiensis, then take cotton, then make them fuse together, then get BT cotton. Then once this new BT cotton is made, I need to get field trial approval, that approval, this approval. It is an easily 10, 15 to 20 years process it is. It will take time. Getting the point here. Here that risk itself is not involved. Because I'm removing it, I know this particular marker is the reason for this. What is it? It's pest intolerance or drought intolerance. I remove it out. It becomes better. It can be used to decipher the host pathogen relationship in crops. Absolutely yes. Consider the following techniques or phenomena: budding and grafting in fruit plants, cytoplasmic male sterility, gene silencing. Which of the above is are used to create transgenic crops? Budding and grafting in fruit plants. Budding means what? Sometimes from the plant, this is a plant you imagine, there will be a small bud which will grow out. Sometimes it will fall off and it will become another plant. That is one thing. Like a small bud will form on it and then slowly from there a new growth will start. Clear? Grafting means you have part of one plant, part of another plant, you attach them together, tie up them together, make them grow. Two different parts, you're making it, putting it together and grow. Some people use this technique to help mango grow a little more faster and better. Alright, those techniques are used by farmers. Cytoplasmic male sterility means the inability to produce pollen. Gene silencing means what? What I told you earlier, mutation. What do you mean by transgenic crops? These are nothing but your genetically modified crops. So which of the above is used to create transgenic crops? Statement 1, definitely it is incorrect because budding and grafting are normal techniques which are used by any farmer. So obviously it is wrong. Statement 2, definitely. Statement 2, if cytoplasmic male sterility means what? The plant is not producing any pollen. Why? Because you have muted it somewhere. You have muted it, or you have made it incapable to produce the pollen. Yes, it can be a method. Systems are widely utilized in croplands for hybrid breeding due to the convenience of controlling sterility. Statement 3 is also correct. And what was that? Mutation. Silence. Gene silencing using RNA interference technology, transcriptional gene silencing, virus-induced gene silencing has been used in horticultural techniques. What we have already seen. As you can clearly see, some of these terms and terminologies keeps getting repeated in different variations or forms. Clear? Moving on. In the context of the developments in bioinformatics, the term transcriptome, sometimes seen in the news, refers to. Transcriptome is nothing but your whole set of RNA, messenger RNA, transfer RNA, ribosomal RNA, plus any other RNA. All of that entire range. B is the answer. Transcriptome is the full range of messenger RNA, mRNA molecules expressed by an organism. The term transcriptome can also be used to describe the array of mRNA transcripts produced in a particular cell or tissue type. In contrast with the genome, which is characterized by its stability, the transcriptome actively changes. In fact, an organism's transcriptome varies depending on many factors, including the stage of development and environmental conditions. That is one point you will have to remember here. It will go on changing depending upon which stage of development it is and also the environmental conditions. Clear, people? All right then, I hope with that we come to an end of our first part. Any suggestions, any doubts or clarifications, or if you want me to take any particular topic of science and tech to make things better or easier to understand, please do let me know in the comment section. Otherwise, you can reach out to me in the Telegram channel. Let's call it a day, people. Take care.